Mic's on?
Yeah.
All right, so, and the camera is,
You're facing that
there we go. So, good afternoon, everyone. This is Ben over at the 5252. It gives me great pleasure to introduce Thomas Mavrakanas from the Division of Nephrology. So he's one of the FRQS junior stage researchers here at CORE, and he's going to be telling us about anticoagulation strategies in advanced kidney disease. So I'll pass the mic to you. I believe that goes over your ear. Yeah. And
hello, thank you very much for the invitation to present you today some of my work on anticoagulation in patients with advanced renal disease. So these are my disclosures. And I would like to talk to you today about the field of anticoagulation in patients with advanced renal disease, focusing more specifically actually on direct oral anticoagulants. This is a field that is now, it's now almost 10 years old. This back in 2010, 2011 were exciting times, not only because we were younger and ambitious, but also because we had every 2, 3, 4 months, one new paper in anticoagulation with these new agents that actually radically changed our management of atrial fibrillation and venous thromboembolic disease. And as you all know, we have the direct factor 10A inhibitors, and we also have the direct factor 2 inhibitors. And we are now having under development the factor 11 inhibitors that are not on this slide, and we may talk a little bit at the end. So you may already be asking yourself what a nephrologist is actually doing with anticoagulation, but my background was a little bit of a staff internist for several years in Switzerland before doing nephrology, and at that time I was working with Henri Bounamont and Arnaud Perrier, who were well-established researchers in the field, and I continued working on this subject in my most recent nephrology days. So these agents are quite remarkable in their efficacy, sorry for the German in the title, and in their safety actually. And as all these big randomized trials showed, they are a decent alternative to warfarin, providing at least similar, if not higher efficacy, and also at least similar, if not better safety. And when you try to extend results from these studies in patients with chronic kidney disease, you start having some issues because actually these agents are eliminated by the kidney in various degrees as your creatinine clearance is dropping below 50. And as we have shown here, the renal elimination may vary from 22% to 80%. And this is something that needs to be seriously taken into account when you are using these agents in clinical practice. So for stage 3 CKD, the key to safe prescription is actually appropriate dose adjustment. And as we have shown in this paper, there is strong evidence. This is the subgroup of moderate CKD from the big randomized trials I mentioned just before. So you see these trials had up to 20,000 participants, and you had several thousand of patients who actually had CKD stage 3. And results are very much similar to what we see in the whole cohort with respect to the efficacy and the safety of these agents. But if the situation is quite well established in moderate CKD, this is not really the case in advanced renal disease. We don't have good pharmacokinetic data. We have only PK data in the subgroup of patients with 25 to 30 mLs per minute of creatinine clearance that were included to Aristotle. And if you look carefully at those difficult to understand graphs, you will see that as kidney function declines, you have a proportion of patients who are getting the full dose of apixaban who are really supratherapeutic. But you do not really see the same effect. If you can see here, you go up to 8,000 nanograms per milliliter per hour, but here only up to 4,000. So the scale is not the same. And you will see that with a lower dose, you don't see the same accumulation that you see here, most patients being in what we consider to be a reasonable therapeutic range for these agents. Unfortunately, we don't have high quality randomized data. There are only a few patients from Aristotle in this window of 25 to 30 mLs per minute. And we only have this randomized trial with another direct factor 10 inhibitor, edoxaban, that was done in elderly Japanese patients, and half of those were actually with stage 4 CKD. And what we see in patients who actually have renal dysfunction is that the hazard ratios and the confidence interval are very similar to those without significant renal dysfunction. So although one may argue that the principle of dose adjustment applies to CKD4, the dose is not yet well established in advanced renal disease. And we have at this time a collaboration with the University of Rennes in France. We're trying to set up a population pharmacokinetic model. We have 21 patients here. There are a few more in France who are receiving actually apixaban for an established indication, so we do not interfere with their treatment. This was prescribed by their treating physician. We will be just dosing drug levels, and we will try to develop a PK model that will also use PK data from dialysis that I will show in the following slides. So this project is ongoing, and we don't have any good data yet to show and share with you. In the meantime, in this position paper, we actually reviewed the guidelines, and you can see that the different societies have contradictory recommendations with respect to what to do with these agents in advanced renal disease. As part of an expert opinion recommendation that you could take with a grain of salt, we are suggesting dose adjustment in CKD4 and careful use of these agents in this population. We warn against the dabigatran because actually this is accumulating, as I showed you before, 80% of renal clearance. So it's not safe to be used in this population. How about dialysis? So atrial fibrillation is an important problem in dialysis, as we showed in this meta-analysis done with colleagues from the UK and David Chariton from New York. We actually pulled in this paper data from patients with implantable loop recorders. And what we're showing is actually 19% of patients have a new episode of atrial fibrillation per year in dialysis. That's 1 out of 5. And 26%, so 1 out of 4, has a new or recurrent atrial fibrillation episode. So this is a common problem in dialysis, and it's associated actually with worse survival and higher incidence of stroke. But stroke in end-stage renal disease is not exactly the same as in other patients with less advanced CKD. This is a very recently published meta-analysis done by one of my fellows in Switzerland. And what we show in this paper is that the relative frequency of hemorrhagic stroke increases as kidney function declines. So 76% of stroke are ischemic in patients with CKD that has not progressed to kidney failure yet. But when you enter dialysis, only 59% of strokes are ischemic. Of course, there's a lot of variability in different cohorts, and these results are not perhaps 100% accurate, but at least we have now some evidence showing that there is a change in the relative frequency of ischemic versus hemorrhagic stroke. And I will show you in the following slides why this is important. So at this point, somebody may say, well, okay, why bother? You have warfarin. You can use warfarin in dialysis. It's established, used for many decades. It's not renally eliminated. You can measure INR. So why do all this work actually with direct oral anticoagulants in a population in which it may not be safe to use them? So unfortunately, warfarin probably doesn't seem to work very well in ESRD. So this is the most recent meta-analysis. It shows actually that the ischemic stroke with or without warfarin is exactly the same in patients with atrial fibrillation who are on dialysis. But the bleeding risk increases by 46%. And the other dreadful complication in dialysis patients is calciphylaxis, which is a rare condition, but this is associated with 50% mortality. So 1 out of 2 patients who will develop this will die. And warfarin increases the risk of calciphylaxis by a factor somewhere between 3 and 13. So I think I convinced you why we need to find alternatives to warfarin in dialysis. And the problem with apixaban that seems the more obvious choice due to its pharmacokinetics, it's actually that none of the randomized trials use this agent in advanced renal disease in kidney failure. And at that time, there were no pharmacokinetic data after prolonged administration. And around 2016, actually, this study was published, and this study influenced a lot prescription patterns. And I'm very sorry to see that it's still being referenced, and I'll explain to you why. So what they did in this paper is that they took 8 or 9 healthy subjects, and they gave 1 pill of apixaban, and they compared this with 8 or 9 participants with dialysis. And they found actually that the curves very much overlap. So they concluded that the exposure to the drug was only 36% higher. And they said actually that they considered that to be modest, and they suggested that apixaban should be used without any dose changing in this population. But this is wrong. And this is wrong because anybody who knows a little bit about pharmacology will tell you that when you have a drug that is eliminated by the kidneys at a certain percentage, this will accumulate over time, and you need several half-lives to see the full effect of accumulation. And what you are getting here after a single dose may not be at all representative of what you will see several days later when you have reached the steady state. So based on this, you see the impact that was published around 2015. It was published 2016, but it was available as a part of the monograph since mid-2014. And you see the explosion in apixaban prescription in the United States following this paper. So when I saw this paper, I was very much troubled, and I designed this study that was done at the Jewish General at that time. So we gave apixaban single dose 2.5 milligrams to the patients. We completed actually the pharmacokinetic curve as the other study did. And then we continued the drug for 8 days. The 8-day interval was chosen based on what is the half-life of the drug. We allowed for at least 5 half-lives, and we expected the half-life to be almost double in kidney failure. So we would have reached steady state by day 5 or day 6 using these calculations. So we got a trough level on day 5 or day 6, and then we repeated the whole PK on day 8. And then we repeated exactly the same curve, but on day 9, and during the first 4 hours, the patient was at the same time being dialyzed. So now you could compare 2 consecutive days without and with dialysis to assess for dialyzability. And after a washout period, we repeated the same experiment, but with the full dose. The other thing is that the study I showed you used mass spectrometry to quantify drug levels, but we don't really know how apixaban will behave in uremic samples. So we selected what is mostly used now, an anti-10A based assay. So the assay that we used is this in red. We'll measure the anti-10A activity and we'll extrapolate the anti-10A activity to concentration. So you are getting a virtual concentration value, but actually what you are really measuring is how much the drug is inhibiting factor 10A. And this is what is most important from a pharmacokinetic and pharmacodynamic perspective. We had established exposure data in the general population with different doses of apixaban, as you can see. So we could have a good reference to compare with. And not surprisingly, we saw significant accumulation of the drug. So this is drug exposure area under the curve. And you see that all patients increase the concentration of at least 50% up to 300%, you see. And we showed also that the drug is not dialyzable. And this was expected because it's a protein-bound medication and proteins don't cross the filter. And the other finding was that with the full dose, actually, some of the patients were becoming clearly supratherapeutic. And this, at that time, the PK data that I had showed you from Aristotle trial were not available. So we're the first to be reporting PK data. But now we know from the Aristotle PK data that we do see some of patients that will overshoot clearly at very high supratherapeutic exposure levels, and this is potentially dangerous. So based on this, we actually suggested the drug to be used at the lower dose and not at the high dose in dialysis. And we recommended a clinical trial with apixaban to be conducted. So the clinical trial happened. It was started the year that we published this, but unfortunately, they didn't take us very seriously, so they gave the full dose. And if you look in the literature, I didn't show you for the sake of time, all reports, you will see that all observational studies in patients with different levels of kidney impairment showed a benefit, statistical or not statistically significant, with apixaban. This is the only paper showing worse bleeding with apixaban. The study was interrupted early due to loss of funding, and they couldn't recruit enough, so nothing is significant here. But they, you know, when you think that you're seeing a horse, even if you see a zebra, you may say still that, no, this is a horse actually, and I have color blindness. Or so this is anchoring. You see, you stick to your initial hypothesis, and if the data that don't fit very well, well, it's probably the data's problem. And this is highlighted even more in their interpretation of the PK. So these data were presented in the European Society of Cardiology Congress, and you will see they are showing very much what we showed, with some of patients going up to 6, 7, 8,000 nanograms per milliliter per hour. And any thrombosis specialist will tell you that this is very high, but they are interpreting this as actually a modestly higher exposure that was consistent with the results of non-ESRD patients. Well, no comments. It's not surprising why this has not been published yet, 2-3 years later. An interesting alternative is rivaroxaban in dialysis. This is a very meticulous work by Anne de Vries from Brugge, Belgium. She actually conducted a very well-designed pharmacokinetic study with rivaroxaban the same time as us. This is half of dose of, so 20 is the standard dose for rivaroxaban, 15 is the adjusted dose in advanced renal disease. And she did the study with 10 milligrams. This is a well-conducted pharmacokinetic study that actually assessed the drug exposure after several days, and she calculated retrospectively the half-life to make sure that she was reaching the steady state, and she found similar exposure with a 10 milligram QD. And then she designed this very nice trial, the VALKYRIE trial. This was a trial where she compared vitamin K antagonists versus rivaroxaban plus vitamin K. And the idea is to actually provide vitamin K as an attempt to stop this metalloproteinase induced vascular calcification process. So this was a translational study. Unfortunately, she didn't show any benefit. She was thinking that probably calcification is very much already established when you reach kidney failure, and that's why you don't see any benefit. But then she followed these patients for 2 years, and although this is a small study, single center, and it may not be easily generalizable elsewhere, she did show a benefit of rivaroxaban compared with warfarin. But now the next question is this really what we want to assess? So if you have a drug like warfarin that actually is not helping against stroke, but is increasing bleeding, then you have your control arm that is actually disadvantaged. So maybe the right question should be a direct oral anticoagulant against nothing. And this was never done until very recently. We did a retrospective study trying to answer this question. So this was a retrospective cohort study using the biggest dialysis data set that exists, the USRDS data set. And we actually developed a propensity score for apixaban prescription versus no anticoagulation. And then we matched patients with treated with apixaban versus no treatment according to this propensity score. And as you can see in the flow chart, we ended up with 1,500 patients without any anticoagulation and 500 patients on apixaban. We had excluded those who had received another direct oral anticoagulant, and we also had excluded patients with valvular atrial fibrillation. We excluded many people who didn't have Medicare coverage in the United States, including Part D, because otherwise it wouldn't be possible to get information on anticoagulant prescription. We wouldn't be confident if they were or not on any anticoagulation. So we examined the apixaban prescription as a time-varying covariate, and we considered death from any cause as a competing risk because of the very high mortality rate in the dialysis patients. So in the matched cohort, as you can see, the two arms were quite well balanced with respect to 23 baseline characteristics that we thought were important, such as demographics, diabetes, heart failure, MI, cancer, liver disease, and several medication. And what we found was actually that apixaban compared with no anticoagulation didn't improve outcomes. So there is no difference in the incidence of stroke or systemic embolism. Something that you want to consider when you approach bleeding in this population is the very high bleeding rate in these patients. So dialysis patients may have up to 50% bleeding rate per year, because actually the widely used ISTH definition is using also the 2 units drop in hemoglobin. And the problem is that when everybody bleeds, you can miss a signal. So we defined bleeding in this study as something that, in our opinion, matters the most, fatal or intracranial bleeding. And as you can see, we saw more bleeding with apixaban compared with no anticoagulation. And then we said, let's look a little bit by dose, because almost half of patients had received the high dose and half had received the low dose. And you will see actually that the risk of any stroke is increasing with the high dose. It doesn't change with the low dose. And you would tell me this doesn't make sense, why your stroke rate is increasing when you anticoagulate the patients. And the answer is here, because actually you get more intracranial bleeding. But at the end of the day, and this was the fundamental error of other papers who tried to answer this question, but in apixaban versus warfarin, the fundamental error was that they looked only at ischemic stroke. But as I showed you before, the relative prevalence of hemorrhagic stroke goes up as kidney function declines. And when you anticoagulate with these patients with the full dose, you get too many hemorrhagic strokes, and that's why your whole stroke risk goes up. And you will agree with me that to our patients, it doesn't really matter if the stroke is ischemic or hemorrhagic. It matters that they cannot move or they are aphasic. So what we concluded was actually that anticoagulation may not be an option in the dialysis population unless you very carefully select your patients. Are antiplatelet agents altering this association? So we just did this. It's in PRESS in Kidney 360. So it's from the same cohort. You will see that almost half of patients were also receiving a P2Y12 inhibitor, most of them clopidogrel, and the other half was not receiving them. And you can see that this is the stroke risk, this is the bleeding risk. So there's a higher bleeding risk in the subset of those who also are on a P2Y12 inhibitor. And of course, it's not surprising. And what is surprising is that still we nephrologists prescribe actually antiplatelet agents in stable patients who are anticoagulated. I will remind you that the FIRE trial in the general population showed that in this population actually that is stable cardiovascular disease, anticoagulated with any agent, you do not need to add an antiplatelet agent if you don't have a recent stent insertion. So in the title of the presentation, there was a mention about lack of clinical trials. So it's not entirely true. There is this study driven from Toronto. It was designed by our colleagues in St. Michael's Hospital. It's a small study, 150 patients. It was looking at apixaban versus warfarin versus no anticoagulation. It's a feasibility study. Recruitment was just completed. We hope to see the results pretty soon. So that's what they said initially. And then we actually, when they approached us to recruit here at McGill, we showed them the PK data, and many other centers did the same. So here at McGill, we used a 2.5 milligram dose. And actually the protocol was modified, allowing for many centers to use the lower dose. I don't know yet how many centers went with a low and how many with high dose, but here in the hospital, in this center, we actually recruited using only the low dose of apixaban. In the waiting, in the meantime, lack of data, but again, the same position paper I showed you before. We are putting this in red, meaning that maybe it's not a very good idea, but if you go for this, because you have a selected patient that you think that may benefit from anticoagulation, you should go with the lower dose, or at least that's what we are suggesting until we have more data. So all this goes about AFib, but how about VTE? And VTE is also a relatively common problem in our population. We know from the literature that actually CKD is associated with higher risk of incident venous thromboembolism. And you can see the relative incidence going up as kidney function declines. So this is, there are two models comparing incidence in patients with preserved renal function, very mildly reduced GFR, and patients with stage 3 or 4 CKD. And you can see that there is a significant trend towards higher instance of thromboembolism as kidney function declines. And this data is reproduced in 3 different cohorts. And what we also know is that if you develop VTE and you have CKD, you have worse outcomes. So you see the mortality rate almost doubles, and also the probability of mechanical ventilation or thrombolysis is higher, especially in patients with kidney failure. So what are the studies showing in VTE? So we summarized here the evidence, and if you look at the hazard ratios, you will see that it's not too bad, with the exception of the bigatran, that indeed is associated with higher major bleeding, but this was expected due to the pharmacokinetics of the drug. But if you look more carefully, you will see that there's a very small group of patients here. And also, if this was designed as a non-inferiority, although you cannot really say that in retrospect, but you will see that the confidence interval is very large, and it may actually be associated with higher incidence either of VTE or bleeding. So although I was quite reassured by the data that I showed you from RCTs in patients with moderate CKD, it's not the same in patients with actually VTE. So to understand a little bit how you treat thrombosis, you need to know what are the objectives of treatment in thrombosis, and we nephrologists don't know that very well. So you have an acute phase that your objective is to stabilize the thrombus, and here traditionally you would go with parenteral anticoagulation. When you use the new agents, you go with very high dose. So this is twice as much as you would give in atrial fibrillation. And this is for 7 days with apixaban, 3 weeks with rivaroxaban. And then you have a maintenance phase, 3 months to prevent early recurrence. And here you use the same doses that you used in atrial fibrillation, but attention, there is no dose adjustment here. So although the RCTs with atrial fibrillation did use dose adjustment for direct oral anticoagulants, this did not happen with apixaban or rivaroxaban. So we have 0 data with reduced dose in patients with any degree of kidney dysfunction from the randomized trials. There is one exception to the rule, this agent edoxaban. What they did in the Hokusai VT trial is that they did not use this drug from day 1. They used a low molecular weight heparin for at least 5 days. And then they renally adjusted the medication. So only edoxaban has shown, and in patients with a creatinine clearance as low as 30 mL per minute, that you can actually, after an initial phase with an LMWH, switch to a renally adjusted dose and have results that are comparable with warfarin. Unfortunately, this was not explored in more advanced renal disease, although there were some patients in ENGAGE and in Hokusai VTE with creatinine clearance below 30. So if you dig in the product monograph of the FDA, somewhere page 120 something, there are some of these edoxaban data, and I'm trying to get actually that data sets to see if we can actually get some meaningful results, because this would be very interesting in the renal population, especially for the thrombosis indication. The other thing I'm planning to do is to do a PK study with edoxaban in dialysis, trying the 30 and the 15 milligram dose. The advantage with this would be essential in thrombosis, because if we show a similar exposure with the drug, we know that the reduced dose works for venous thromboembolic disease. So we would have perhaps another treatment option for our patients. Last few minutes will be dedicated to the future. So if we go back to your physiology textbooks, you will find out that actually factor 11 is crucial in the activation of the endogenous pathway. contact phase with collagen. And the thrombus growth and stabilization depends on feedback activation of factor 11 by thrombin, which amplifies thrombin generation and fibrin formation. On the other side, the role of the factor 11 in hemostasis is nominal. So inhibiting factor 11 may be an interesting therapeutic approach to activate only the contact pathway and actually use this in patients such as hemodialysis who are at very high risk of bleeding, and you want your hemostasis process as intact as possible. There are some phase 2 trials with these agents that are not yet available in the market, using them as an anticoagulant for the filter in hemodialysis. But we don't have data on hard outcomes in patients with reduced renal function. But maybe in 10 years from now, if we do this talk, you will see this slide as you were seeing the first slide when I was telling you about the exciting days back in 2010. So maybe there are more exciting days ahead. So to summarize, chronic kidney disease associated with higher risk of incidence venous thromboembolism. Patients with venous thromboembolic disease and CKD have worse clinical outcomes. The direct oral anticoagulants may be considered in moderate CKD, but unfortunately, we don't have high quality data, that's the word missing here, in patients with advanced CKD, especially in the thrombosis field. And the factor 11 inhibitors may revolutionize management of VTE in ESRD. And I think at this time, presentation is over and I'll be happy to take questions. Thank you very much. Sure. So if I repeat the questions, yeah. Oh, perfect. It's okay. Yeah, it's fine. Yeah.
There are probably a lot of drugs that merit this kind of careful study in the dialysis population. I'm thinking, for instance, and this is a thought that crossed my mind, of SGLT2 inhibitors, where they slow deterioration in renal function. We're seeing more and more, even at doses where they don't impact the glycemic levels. And I had a patient recently who was on dialysis, but has proteinuria, has Mavrakanas glomerulonephritis, and is on an ACE inhibitor. And I was wondering, maybe SGLT2s have a role here. And so, just your thoughts on
So you're touching a sensitive chord, because actually we just got approval by the ethics committee here to conduct the first pharmacokinetic study with canagliflozin in dialysis. So what you're suggesting is very interesting, not only because some of patients may have residual renal function and benefit from the renal effects of the drug, but also, and we're getting out of anticoagulation, but we stay in the same methodological field. But some people may get benefit at the heart level. There is there's a hypothesis about the hydrogen NH3 receptor at the heart level that may be affecting the cytosolic concentration of calcium. So you could have some cardioprotection that is independent of the renal effect of the drug. So you could imagine using these agents even as actually cardioprotective drugs in patients with zero kidney function. So a first step would be to make sure that you will not get very high exposure. And that's why we used canagliflozin, because it's 99% excreted outside the kidneys. So we picked up the less renally excreted agent because DAPA is a bit more, and EMPA is closer to Cana. And we are actually trying to do the same thing as we did with apixaban and compare with established PK levels that we do have in the general population and see how far we go to account for potential toxicity.
quality of evidence for the benefit of DOACs and AFib in the context of dialysis is relatively weak or poor quality of it. We did the PK studies that were nice. We did this pretty large-scale propensity score weighted observational study that showed, not much benefit, really excess harm. Is that fair to say? Like excess death is related to hemorrhagic stroke. I would have stopped there. I mean, I guess I would have said, because initially I was like, well, maybe we should just do a trial, but we did the right thing, which is, well, let's do the propensity score weighted observational. And it seems like there's excess, recognizing the limitations of observational data, propensity score weighting, perhaps some unmeasured confounders that we're not addressing. Nevertheless, you see this excess harm, certainly not excess benefit. And so then I wonder what the value of the trial is. And maybe that's why you're not leading the trial.
Yeah.
It's hard to, I guess you suspect that maybe there is some problem with the propensity score analysis.
Yeah. So, to summarize the question in a few words. So the question was that actually there is the PK study that's showing actually the accumulation of the drug. There is also the evidence for the propensity score study that shows more harm than benefit. So why bother doing a clinical trial in this setting? So I think you're probably right. And if I had designed safety, I would have done it differently. But I think there is no harm signal with a low dose, the 2.5 BID. We didn't see excess strokes there. And because we know that warfarin is associated with harm in dialysis and we have many pathophysiological mechanisms going in this direction, so one may argue that actually you could try a low dose direct oral anticoagulant trial in this population because the propensity score doesn't show much harm. The PK is showing that probably you are where you should be. And then you never know with observational data, even with all the possible analysis, still the quality of evidence is relatively weak. So perhaps with a low dose of the direct oral anticoagulants, that could be a potential study, although I suspect that if factor 11 inhibitors continue at this pace until, it will be probably a question of the past, the factor 10A inhibitors in this population.
Follow-up question, thrombosis. Would there be any value in using that same data set, the US registry, to explore the same, to explore the outcome of VTE?
So actually, the question is about using the USRDS to ask to access the data set and have the question, answer the same question on VTE. So what we, it was just published, actually. It was just published. I was the reviewer and wrote the editorial. So it shows that there's a trend towards some benefit with apixaban, but it's still, apparently they tried as much as possible to identify a thromboembolic episode close to the apixaban prescription. So they believe that they did their best to select patients who are treated for VTE. Of course, retrospectively, you never know. But the paper was published and actually in CJSON a few months ago, and shows a small potential benefit from apixaban in this population. Yeah. Yeah.
Problem is you also have your really compelling pathophysiology that the hemorrhagic strokes
Exactly.
are a bigger problem, which is actually the first time that message has sunk in. So that's concerning.
Yeah. So the
I'm not going to prescribe them in any dialysis.
So the comment by Dr. Dasgupta is actually the pathophysiology about the increasing prevalence of hemorrhagic stroke as kidney function declines that makes even more dangerous use of these agents in this population. Yeah.
I'm encouraging them to ask questions, but they know what.
Okay.
Shy, very shy.
Okay.
clarify, but I'll go ahead.
Sure. Mhm.
I haven't read it properly yet, but there was a recent McGill study that came out in rheumatic heart disease, valvulopathie patients, suggesting better outcomes with Coumadin than with the DOAC. I don't even remember which one.
So,
And then there's the previous sort of anticoagulation studies also suggesting that. And it surprised me greatly because you know what you're getting with the DOAC, at least in terms of anticoagulation, whereas with warfarin, they're in and out.
so the comment by Dr. Dasgupta is about use of direct oral anticoagulants, either in patients with antiphospholipid antibodies or essentially in patients with metallic valves. So you're absolutely right. You're alluding to the real-line trial probably. And it showed definitely that outcomes are worse with direct oral anticoagulants compared with warfarin if you have a metallic valve. And the same was actually the result of the study with the patients with antiphospholipid syndrome. So I think that especially for the metallic valves, maybe it's a more complex problem. You see the contact, the propensity to develop a thrombus when you have a foreign body, and the activation of the different contact pathways may require a more comprehensive actually anticoagulant that will block several factors, because that's what you do with a vitamin K antagonist. So this is probably the explanation behind that, but I agree with you. Probably, in my opinion, the only solid indication for warfarin in dialysis or even in the general population is a metallic valve.
Shown as, and you can repeat this comment because it's a compliment.
Oh.
Having physiological expertise, at core, joining that was really important to pathophysiological understanding. And so, really thanks for being here.
So thank you very much for the invitation and for your kind compliment about the epidemiological and pathophysiological studies. And I'm looking forward to presenting here again, perhaps with respect to SGLT2 inhibitors, we'll have some results. You're welcome.